Okay. So our next speaker is Xinyang Zhang from Georgetown Washington University.
Hi everyone, my name is Xinyang and I'm a second year PhD student from George Washington University and I work in Dr. Ran's lab. So today I'm going to introduce a bioinformatics tools called seqSight. Well, the seqSight is called like the profile, a jointly profile microbiome strings, genes, and biosynthetic gene clusters from metagenomics data. So before we get into what seqSight is, we can go a little bit quick about evolution of taxonomy profiling tools. So here is many of the tools which are like well known and very good to use, like Periscope, Metaflow, and Kraken. I know some of them already for version 3 and version 4. So, and I use the most of them in my researches, and I think they're very good, like a very good, like a mapping accuracy, and they're very comprehensive to use. And the more research, like I apply them into, I find that what the people are looking forward for a tour. And I think they really want to find a tour that can do a very comprehensive analysis framework. And also they have like a ability to handle ambiguous rates, because like it's very important to you if you have a multi-map and you want to level up the accuracy. So let's go into the seqSight. I know this kind of like page is kind of our ambitious word, but today I'm going to only focus on what is the core module here. Let's see from this top left, which we take our shotgun sequencing reads out our input. And we do the filtering and the QC module, which you like separate your host reads and your microbiome reads. So based on what's your target, what you're interested in, and we go our main part is called microorganism mapping. So what's the important things here is like when you have their reference database, if you have their mapping strategy, and everything can be fixed. And here, after you have your like a mapping strategy table, and we can do some downstream analysis like alpha diversity, pathway analysis, or the more interesting part here is called the metabolomics and BGCs relationship. So I know like I guess you might have a very like a little bit of impression about what the seqSight is. Let's go with the one example about how this looks like. So here's one example from COVID-19. And we all collect the samples from COVID-19 patients, and they're all nasal swab samples. And we sequencing them and got the FASTQ file or the FASTQ file. And we have our 5 reference database like human, fungi, archaea, bacteria, and virus. Well, since the COVID-19 is the kind of like SARS-CoV-2 is a virus, and we want to target on the virus only and separate the rest of 4. So what do we get? We got the result A and the result B. The result A is called abundance table, which is the potential like virus genome ID there and their like value here. And B is called alignment rate, which means like how much % of your reads can be mapped to the different reference database. So what we can do based on these 2 kind of type. The first one is like if we have abundance table, we want to do some taxonomy profiling. And the one thing we can do is that we can use their NCBI's officially API called Entrées to do the mapping. But I suffered a lot because like it got so many limitations. Like if you can only request like 3 times per second or 10 times per second, it's never end. So in the end, I think, why not I just build up my own reference database, which contains all of their like bacteria's ID, bacteria name, and all of their information, which will give you a more quick and more easier answer in the end. And also for the result B is alignment rate. And when you're like multiple accumulate of your samples alignment together, we can have a QC graph. We call them QC graph. So in the bottom, it will show you like the distribution of this 5 reference database. And in the top, it show you like the reads from each sample. So when you got this graphs, you might think like, oh, whether this a big proportion of this human race is you wanted, or you want to trim a little bit, or you want to do anything that to fix your sample. So I mean, this graph can give you a very like the good reference for this. So after we're talking about the species taxonomy reference database, we go more interesting part. It's called the BGCs part. Well, I don't know, like many of our guys know what this BGC is, but I'm super new to this field, but I will give you a brief introduction about what BGC is. So BGC is called biosynthetic gene clusters, and they are locally clustered groups of 2 or more genes that together to encode a biosynthetic like pathways, which is like have more functional. So what they're functional, for example, they can interface the computation with their surrounding microbes to reduce the the resources are very finite, and they can be interface the computation with that. Also, it can provide a host cell with resistance against the cell-produced antibiotic. So there are like many different kinds of the function we can apply. So I mean, that is like the BGC works and what we care about is. So after I mentioned before, the mapping strategy needs your like a reference database, needs your like the mapping strategy. So we go a little bit about the math part. I know this kind of thing. I hope it's not that boring. So when we talk about Bayesian reassignment model, like we talk about stats, we always go first with the assumption, right? So each read, like we have the input reads here, like each read will be mapped to at most 1 BGC in the reference database. We know the BGCs is kind of lack of annotation, it's not like everywhere. So but what's the reality is like it depends on how conserved that region of BGC that it will show some multiple map. So which one is the correct match? Let's see example. So here we have our input rate and it is our reference database. We can see that the pink one can be only find there's 1 genomes in there, like 1 BGCs in the reference database. So we'll give them the 1. There's only 1, right? And this thing that the second one is the red. Okay, it's got 2 matches. It's okay. We take them like 2. It's just like count how many like matches there. So the same idea for the green, still the 2 matches. And the blue, okay, we have only 1, perfect. And in the end, we're gonna find there's like one which against our assumption, which is like a multi-map. So based on this, like the multi-map, we need to make a decision which one you want to use and which one is like give them correct value. So we will bring another topic called mixing data mixture model. It's okay, I know there are so many formulas on camera here, but I will give you guys a take home that is like we were not like equally give them a 50 like the equal chance. We were based on their proportion and give them the bonus or the penalty to reassign them a new value of the abundance value. So you might ask like how this work? Is there like good enough? So we do a simulation study on comparing like different tools on the market. And in the top is the yellow light bar, which represent our Bayesian reassignment model. And I think in the top, like 4 chunks here, it represent different lengths of the read. And they all performs pretty good in different lengths. And interesting part, like I know the MetaFlam performs also pretty good as well. And also they have a very fast running time. So it kind of depends, like if you know your read is quite long enough and you want to have very fast, you can choose the MetaFlam, or you don't know like which kind of lens you have, so you can choose the Bayesian reassignment model. So after we know that it's very like good to use and very high quality, we want to go another step into another application. It's another research. We want to find the relationship between the mother breast milk and infant gut microbiome. So what do we find? And we want to find whether they share the same microbiome with each other or with the relationship between the metabolomics and the BGC. We know that the BGC can come from the metagenome after the seqSight running. And we know that there's some of infant, like the omics feature about the metabolomics, and we want to find that interesting thing. And there's also a poster in our lab. If you're interested, we can go into that. So I know it's a huge, I think it's so many information. Here is like we quantify the BGC abundance in our samples, and we see the sample cluster pretty well by type. Like for the green part in the top here, it represents breast milk, and in the blue part it represents infant stool. So it's like the cluster by type very well. And we also do some of these or additional ordination plots, and we see that samples cluster well by the BGC abundance, like which in the bottom, and the cluster are very comparable to ordination based on species, and we use the MetaFlam. So, but the why we're interested in BGC is that even though they share very same pattern and they clustered even the same, in case just because they're different directions, it's okay, but they're still clustered very well. But their BGCs has got more functional, and which is this is one more benefit we want to interest in them. So let's jump more deeper. So here's another graph that counts like how many metabolomics feature can be related to your BGCs. So some of them is like a topic has a very long tail, and some of them has like over like 300 of their metabolomics features can be related to one BGCs. So that's kind of an interesting part. And we do the Spearman pairwise correlations here. And we found interesting study, and we try to find their like top 8 strongly positive correlated with the deuroblin and the Acinetobacter. And you know, like deuroblin is a byproduct of hemoglobin breakdowns. So I mean, as I mentioned before, this is still undergoing project. We are still trying to investigate what's the significant findings here. So in the end, like I want to quick wrap up here, and the seqSight is the kind of a tour, a platform for you to run the things to find the BGC profile taxonomy things. And yeah, if you have a reference database you can also use our database profile for you. So I mean, in the end, I want to express my grateful to my Dr. Renovat and my like co-workers like the Tyson Dawson, who give me great help. And they all both have their mentioned 2 researchers, and they all have the poster section. So welcome you guys to have a seat to talk with that. Thank you so much.